Hello, everybody. We are thrilled to be a little bit of a transition panel. We are talking about drug development in rare pediatric and adult populations. So we'll do a little bit of matriculation to the adult space. I'm so thrilled to have these panelists with me for the discussion today. If you can go to the next slide, there's actually a definition on this slide of what mechanism-based drug development is, because I think it's really important to ground the session in what we're focusing on. And the curation of this panel was to bring the perspective of mechanism-based drug development, but to cover it holistically from different segments of the drug development ecosystem. So as we go through our discussion today, you'll see that we have a representative from a nonprofit research organization and advocacy. We have some people with experience in actual, hands-on drug development, representation from the regulatory space, and a clinician researcher who has worked on developing diagnostics. So we're going to cover mechanism-based drug development. There will be a quiz at the end. No, there won't. We'll have question and answer at the end. But right, and no diagnostic testing at this panel. But I do want to make sure that we all kind of hold in our minds while we go through the discussion today that what we're talking about here is really focusing on understanding the underlying biology of the disease so that we can have a rational drug design that targets the mechanism so that we can move beyond trial and error and hopefully have more success in designing clinical trials. So we'll talk through what the needs are, what the challenges are, and these panelists will all help us appreciate that from their diverse perspectives. So with that, what I would like to do is maybe just start with a couple of questions. And obviously, we're going to be hearing from every panelist. And I'm going to start with Todd. I know that you've done really important work and have shown some recent progress in developing a diagnostics. Could you just share a little bit about how that came about and why it's so important and how it's actually already changing clinical trials in the space?
Yeah, so my background is a neurologist, and so I spent the last 30 years really treating patients in the clinic with neurodegenerative diseases, and that includes Lou Gehrig's disease, Alzheimer's disease, and Parkinson's disease. And so when I had just completed my training, the 1990s was labeled as the decade of the brain with a lot of enthusiasm. And as you are aware, we don't have really many breakthroughs for those three diseases. And when I give a lecture on ALS, I usually put up a slide that shows all of the trials that have failed in the last 30 or 40 years. And it's sort of embarrassing because I was a part of those trials, and they include literally the trial and error that you mentioned, which is throwing ceftriaxone, an antibiotic at ALS, throwing lithium, an antipsychotic at ALS, throwing minocycline, an acne drug at ALS. Basically, if you had a drug and you had an untreatable disease, you threw a drug at it. And not surprisingly, that has not really led to any breakthroughs. And so when I've sort of looked back at that career, which has been frustrating for me, I actually kind of asked myself, let's look at where really major breakthroughs have happened in the last 30 and 40 years. And for me, I think about HIV and cancer as being those breakthroughs. And so if I go back to my medical school time, when HIV started, everybody died of HIV. And basically two things happened that now allow HIV to be a chronic livable disease that everybody should do fine with. And those two things were a biomarker, so that we could measure the amount of the virus and that we could measure that repeatedly, rapidly, and consistently. And then that allowed pharmaceutical companies to throw a ton of drugs at it. And what we learned about HIV is that not one drug treats everybody with HIV. Different people need different drugs, and over time they might need different drugs, but because you can constantly measure the viral load, you're able to deliver precision medicine. And so it's that combination of a diagnostic and then broad therapeutics. And obviously the explosion in treatments in cancer is the same, right? 50 years ago, everybody got the same chemotherapy for breast cancer, for colon cancer, for whatever. We now know that that's stupid because we can characterize each individual's cancer based on the proteins that they exhibit, and then we can target therapies, and we treat patients individually. And for me, as someone who's treated the neurodegenerative diseases, we have failed on both of those fronts, right? We have not had a biomarker, so therefore everybody with what we call Alzheimer's disease is lumped together, even though we now all believe it's a very heterogeneous disease. And so many therapeutics that might have worked for a subpopulation of patients that are called Alzheimer's disease fail when you lump everybody together. The same for ALS, the same for Parkinson's. So until we can really get to the place to say we can define, let's say, a person with Parkinson's disease based on the proteins that they're accumulating, and we now know that there are different proteins in different forms of Parkinson's disease, we're never going to be able to treat that disease accurately. So I founded our company about 10 years ago with two other neurologists up at Harvard with the idea that we could use the nerves in the peripheral nervous system as a window into the central nervous system. So the problem with the brain is you can't keep biopsying the brain and ask whether someone's Alzheimer's is getting better. That's not going to be very practical. But we've been able to show that at least the protein that accumulates, for example, in the majority of people with Parkinson's or the majority of people with dementia with Lewy bodies also accumulates in the peripheral nerves. And a little skin biopsy turns out to be a very simple way to then analyze that protein. And so we brought that to commercial testing about 5 years ago. We've now had almost 50,000 patients in the US be tested and help with their diagnostic journey. And we're partnering now with pharmaceutical companies because we've also developed through an AI modeling system a way to quantify the amount of that protein. So now you could treat a patient and over 6 months ask the question, did the patient clinically get better? Did their protein levels change? And really try to have a much cleaner look again at that personalized medicine, hopefully identifying subgroups that respond even if the whole group doesn't respond.
Thank you so much for that. I want to come to you, Dima, because you've had some experience working in drug development in the rare disease space. Based on what we've kind of introduced so far in the panel to this point, can you articulate for us how if you're trying to do the actual work of drug development, how does it change the way you approach it when you're targeting mechanisms? So that, Todd helped us understand the importance of the diagnostics, the confidence in putting the right people in the trial and measure the right things. But how does the mechanism-based approach apply when you're, when you're trying to develop the drug? How do you choose which mechanism to go for? And how does that affect your clinical trial design eventually?
So I have two experiences, one with success and one with failure. And the failure ones are actually what teaches us something. So in 2002, I was working with Genzyme, and we had in the span of 10 years, we only developed 4 drugs, and all of them made it to the market because it was an enzyme that's missing. You replace this enzyme, and the patient lives. It doesn't buy them time backwards, but they live from here onwards. And it worked. These patients who lived afterwards, they do have like another disease now, a newer disease that looks different, behaves different. The patients are aging and they need other supports. At times it's available, at times it's not available. Now, on the other part of my experience with the failure, and only 2 days ago, I heard of another failure, which is very unfortunate in the mitochondrial diseases. So similar to lysosomal storage diseases, they are all inborn or inherited metabolic diseases. And in one side, we had the success, in the other side, we didn't. And I mean, I know I should not be speaking about others, but I can tell you that there is a lot of similarities, and in most of the cases, I still didn't analyze the last case. It's because the mechanism of action of the drug is actually incompatible with mitochondrial diseases. So not only are we treating the wrong disease, we're using the wrong endpoint. We know nothing about the biomarkers. We don't know how to diagnose those diseases before there's an irreversible damage that happened. And then we are actually, say, you give food to the mitochondria, and the mitochondria doesn't work, so you just accumulate food inside the mitochondria. It's counterproductive. So this missing link between the mechanism of action of a drug and the end result of a clinical trial that became a bit bigger, went through so many missing checkpoints to have 4 different drugs with 4 different companies, with number of patients that don't have a day more to waste, traveling across the different countries to get that treatment, being in clinical trials that are very demanding, and then having to deal with the trauma, not only of their disease, but of a failed hope that they had. So, and I think that probably not the right time to discuss the reasons, but I think if we work together, all the 4 different companies, with all the patients that were involved, as early as possible, had the conversations with the FDA, use the platforms that are available in the Critical Path Institute, I think we would have used those mechanisms of action to treat other diseases and maybe prove the concept in other diseases and then prevent the diseases from even starting, rather than wait when the damage is irreversible. Until this moment, we cannot reverse cell death.
Right. And I think, so thank you for sharing those perspectives, because it also helps reinforce, you know, the complexity. We're talking about very complex diseases, maybe heterogeneous presentations, multiple mechanisms could be at play. This is why it's really important. There's kind of like an evolution going on in drug development to, and it's something that the FDA, you know, certainly advocates for, is to identify disease staging that's based on biology, so that you can understand which biology is changing in the early part of the disease, which biology is different in the middle or the end of the disease, so that you can potentially have a different mechanism-based approach that's appropriate to the disease stages, and then also use the drug development tools and endpoints that are appropriate for that disease stage. So that brings me to a question I want to come to you, Gumei. As you know, you are, you know, at the FDA, when you're evaluating a drug effect or a clinical trial that is mechanism-based, how does it change it a little bit? Because you're needing to look at safety and efficacy, but also proof of mechanism. So can you just like share with us how the agency sees mechanism-based evaluations?
That's a great question. I think there's mechanism-based treatment, but not all mechanism-based treatment are the same. And from the failure example that Dima mentioned, I think she brought out a very important point. That is, it's for a mechanism-based treatment to be successful, we need to really understand the pathophysiology of the disease. And in that context, we need to understand the mechanism of the drug, and also whether or not that drug addresses a root cause of the disease, or at least targets a major driver of the disease pathophysiology. So that's a very important thing that we want to see the data to support that mechanism of action. In addition to that, I wanted to talk about the gene therapy in specific, which is almost a poster child for mechanism-based treatment. There are several considerations that are quite unique to gene therapy. One is the vector safety, and in the context, I'm specifically talking about the viral vector-based gene therapy and delivery of vector, particularly in the diseases that affect the central nervous system, like ALS, Parkinson, Alzheimer's. We need to think about across the blood-brain barrier or direct delivery into the CNS, which is quite invasive, and require really unique monitoring and skill sets. And the other thing about gene therapy, when I think about is long-term follow-up, is the the safety of the vector in the context of long-term follow-up. What do we expect to see? And what kind of monitoring should we have? What kind of approach? I think there were a lot of discussion about this earlier today. And what can we do in terms of develop a a least burdensome patient-centric approach for long-term follow-up?
Yes. Yeah. Thank you so much. Andrew, I want to come to you for a couple of, again, as I mentioned, we're really working on capturing the perspectives across the drug development ecosystem. So you are not a doctor. You don't even play one on TV. But you and where you are at the helm of the National Ataxia Foundation, I know, and I ask you to share with us a little bit more about how the National Ataxia Foundation engages the community. And like, for example, you have sponsored a natural history study that is helping us to better understand progression of spinal cerebellar ataxia and other rare inherited ataxias. So I would like you to talk about that a little bit, but also something that you and I have talked about before is that when we come to the example of spinal cerebellar ataxia, so this is a rare inherited neurological disease. It's not pediatric onset, so this is why the adult part of the panel here. But one thing that's very complicated about it is that there are many different subtypes, but you engage everyone in the spinal cerebellar ataxia community. There are some drug developers who are making a mechanism-based approach that isn't a gene therapy, so that might help all ataxias, but then there's other approaches that are more individualized or nuanced to the individual diseases. So could you just explain like how you engage the community and have the conversations as someone who isn't their doctor for where there's activity and hope in the drug development space?
Sure. Thanks, Terina. Thanks for having me. So the first part of your question, how do we engage the community? So, I mean, the easiest way to describe this maybe is literally our mission statement, right? So we have two parts of our mission statement. We want to accelerate the development of treatments and a cure. And to me, the easiest way to describe that is we are trying to make this ataxia space as attractive as possible to drug developers. So sponsoring the natural history study, right? I don't think there's any question that natural history is an incredibly important sort of basic tool, especially in the rare disease space, especially in slowly progressing adult onset diseases. It is essential, and we're seeing that in new drug applications to the FDA using natural history as a comparator set. You're seeing more and more of that, and I expect and hope that will continue. So that's just one example, for instance, of trying to make the space as appealing, if you will, as possible for biotechs to, when they're looking out at where they can go, ataxia is a space that we want them to consider. The other part of our mission statement is to support patients and families living with the disease, and we do that in a whole bunch of different ways. But to be honest, we joke as a staff a lot. I mean, we want to put ourselves out of business, right? And we're a ways off from that yet, but even in the last 10 years, there is a lot of exciting developments in terms of the research pipeline and drug development. The other thing I will mention is, so yes, the National Ataxia Foundation covers all of the ataxias. There are close to 200 hereditary forms of ataxia, and they're discovering more frequently, as well as all of sort of the acquired or environmental ataxias. We cover them all. Maybe not surprisingly, I think the majority of our resources go to those forms of ataxia that are most prevalent. So even combined, the ataxias are a rare disease. Some of them are ultra rare, but some are not. And so not surprisingly, it's where most of the investment has gone. And we do believe that unlocking some clues in one form of SCA is going to have benefits across the others. But we also, again, and as you can imagine, we're spread fairly thin as a foundation. We have funded, I think, research in 60 different types of hereditary ataxia, and not just in the last, say, 10 years. So we don't We want to make sure that we include some of the ultra rare, but clearly most of our resources go toward those types of ataxia that are more prevalent.
That's great. And I know also that, you have many roles that you play at the National Ataxia Foundation with the community, but you also have a big component of educating the community. And Todd, I want to come to you, pulling a little bit more at the education thread in terms of when you wear your white coat and you encounter people in the clinic, do you think that what more should be done and who should be engaging the community to help them understand these nuances in mechanism-based drug development, that this isn't the old playbook? Where do you think more could be done?
Everywhere.
Everybody, all the time.
Yeah, that one's easy. I think the theme actually, that both Andrew and Dima brought up is an important one that I didn't mention. So a lot of these diseases that we're talking about for kids or for adults are progressive degenerative diseases. And the likelihood that we're going to reverse any of that in any of our lifetimes is probably zero. And so as Dima pointed out, the goal really is to stop the progression once you identify the disease. And the ataxias are great examples, right? So people are born with the genetic abnormality and have their first real symptoms in their 5th or 6th decade. That disease is clearly developing for 50 or 60 years. We now know that Parkinson's probably starts at least 10 or 20 years before the motor symptoms begin. Alzheimer's at least 10 years, if not many, many more years. And so I think one of the big pieces of education is really this concept of disease identification, and in particular, early disease identification. Because again, if we wait until somebody, again, has Parkinson's disease and we know that they've lost 60% of the nerves in their substantia nigra before they develop a motor symptom, it is way too late to think that a therapeutic is actually going to have a big effect in that disease. And so I think, unfortunately, it's not as glamorous, and we've seen obviously big breakthroughs just recently with the FDA indication of the first blood test for Alzheimer's disease. But moving all of that to earlier and earlier detection, I think, is going to be another really integral part of ever having a major effect in these diseases. And again, Alzheimer's is a great example of that. We now have drugs that can lower amyloid in the brain by 80% after a person is symptomatic. And the clinical benefit is just not very dramatic. It's just too late to do it. And so I think the idea of, screening at some level, finding some way to say there is a neurologic process earlier in all of these diseases is going to be critical.
Absolutely. Gumei, I want to come to you for something that's actually something we talked about earlier. Everyone at the regulatory agencies, of course, sees a ton of data, and as we've heard recurring through the day-to-day, every individual drug trial, every disease is an individual case, and it needs to have an appropriate case-specific approach. But can you help us understand, one of the things that I think is sometimes difficult for the general community to understand is how you evaluate what we were talking about earlier, remaining uncertainty, in the sense that if we've talked earlier about a gene therapy for severe early onset, rare pediatric diseases. There's the uncertainty is, how long does that last? Certain questions there. But how do you all view the complexity of the remaining uncertainty when we're talking about potentially intervening or preventing the diseases when we know that they can be genetically diagnosed? Can you just, share a little insight there?
Yeah, I think it's contrary to the common belief that we actually do accept certain uncertainty. And I think the key point for us to accept uncertainty is with good quality data, and show us the data, show us the rationale, show us the treatment effect, the safety profile. Then we'll discuss the uncertainty and see whether or not it is acceptable. And could I just make a comment to Todd? I mean, you talk about education and the conversation. I think it goes both ways. And in the case that, in the context that we're talking about prevention of a chronic disease or early intervention at a preclinical stage or early clinical stage, We want to hear from our patients. What matters the most to you? What is the most important to you? What kind of treatment effect you're expecting? What kind of safety adverse events you would accept in this context when you do not have any symptom yet? So that is very important to us. And so we want you to talk to us to really educate us in terms of what your needs and what are your concerns.
Absolutely. And I want to come to you, Andrew, to further the point that Dima, everyone's made, Dima, Todd, Gumei have made. But coming to that part, I know that the National Ataxia Foundation not too long ago had a listening session with the FDA. And I know that, you, of course, as we talked earlier, engage the ataxia community so you can be a conduit of understanding what matters to the individuals and what is meaningful. Could you just share a little bit about that experience?
Yeah, so we did both the patient-focused drug development meeting that is a sort of a formal process. Did that back, I was behind a fiberglass shield. It must have been during COVID. But then we also did a specific patient listening session for spinocerebellar ataxia type 3, which is the most prevalent of the SCAs. Had a group of, I don't know, 5 or 6 patients who agreed to sort of share their stories. This was all done virtually, but had, I think close to 50 attendees from the FDA, which was terrific. And I think one of the really interesting things that came from that discussion was these patients sort of had filled out some surveys about their symptoms and their quality of life and the things that were most important to them. Pain, the fact that many SCA3 patients live with chronic pain was sort of new to, I think, certainly the representatives at the FDA, but even some of our drug developer partners, in not necessarily something that they had considered potentially as endpoints, et cetera. And so just from that discussion, I think, the group that matters here learned something. And, I think for us in the ataxia space, and Todd, you mentioned the other movement disorders that I think are maybe perhaps better known. I think one of the roles that we really can continue to play is that education, not only obviously for our patients, but for our regulators, in helping them understand what's going to matter to these patients and thus where a treatment can be effective.
And that's really important because we're talking about rational drug development here. That also requires this sort of rational engineering, forward engineering of drug development tools. Because once you have better understanding of what matters, then we can start looking at, are there unique mechanisms within spinocerebellar ataxia type 3 that are driving this neuropathic pain symptom, and then tie that to a pathway, tie that to a target. Now we have mechanism-based drug development. I'm that oversimplified, but that's really the idea. So I also want to come back to a recurring theme that we've talked about on this panel, and we've also talked about it in previous sessions at this meeting, is that the importance of data sharing. And I want to also make a plug, I forget which session it was in the morning, where we talked about the importance of sharing qualitative data. And I think that groups like the National Ataxia Foundation, so where you're interacting with the community on a regular basis, can be a really important conduit for that. So I think that's really important as well. I think in the spirit of continuing the listening and the education, I want to ask anyone in the audience if you have questions about mechanism-based drug development, anything specific for our esteemed panelists. Please feel free to share your thoughts. Looks like we have a taker.
Hello? Oh, great. Thanks. Just following up on the earlier session that was dealing with preterm infants and the lack of drug development in that space. I'm actually trying to develop drugs in that space. And this whole idea of mechanism-based drug development in preterm infant population, there are multiple disease states that are all driven by a common underlying mechanism. If you're trying to develop a drug that targets that mechanism, does it make sense to go after a composite outcome score as an endpoint? Or would it be from a regulatory standpoint or from an industry standpoint? I'm looking for advice here. Or does going after sort of one indication a better clinical development path? I know that's a big question, but
Yeah.
Well, it depends. So,
you
I
can't
think
say
it
that
really-
depends. No, sorry.
Go ahead. I thought you were going to say something else. Okay. I think it really depends on the drug development program, on the disease, on the stage of the disease, on what you're trying to measure at that stage of the disease. Obviously, during the duration of a clinical trial, which could be 1 to 2 years, and what you're going to measure at a preclinical stage versus what you're going to measure at a, say, late stage disease are certainly different. So my advice would be come talk to us. We have many mechanisms of early interaction. Come talk to us. And there's no time that is too late to talk to us. So at any given time, if you have a question, it would be great to start a conversation.
Can I say something, Terina? So I think following up on the point and on the question, I think I have a little bit some comments. The first one is in human, there's not one size fits all. To have the same birthday in this room, while it's a very common thing to be born, it's very rare to find 2 people in one room born on the same day, the same year. So imagine with a very rare disease. The same thing applies to that rare disease. Now, every patient is very different. And this is proven from the days of diabetes where it was a killer. And therefore, any disease that kills a child at the early stages of life is rare until there's a drug that allows them to live longer. And now we know a lot about diabetes, and I hope we will learn about rare disease quicker than hundreds of years of diabetes knowledge. And because of the fact that the discussion with the FDA is not happening early enough, this is causing an IND for a drug that is stuck in an indication. And therefore, we have a lot of shelf drugs, like those 4 failed in mitochondrial diseases now, that they will not have the funding to do it. There's a lot of reasons for them not to try another indication now, because the IND was made not on the mechanism of action. Right. And the idea is not very novel, antipyretic, antimimetic, immunosuppressant, anti-inflammatory, painkillers, all of them are mechanism of action-based drugs. So it's not a very novel idea. It's just now the mechanism of action is a little bit more complicated, is more fine-tuned. When it comes to metabolism or metabolic diseases that are like the majority, not the majority, like 4,000 out of the 10,000 rare diseases, that's another system that is very fine-tuned. And more than 4,000 pathways are interconnected too. So any change is a stressor on the organism that, without really understanding the mechanism of action before you embark in an indication and link it with and also understanding the pathophysiology of a disease that is one patient with Mila's case from this morning. So you cannot build a natural history study when there is one disease, because we know that the second patient will be alive, will have a very different story.
Right, right. That's a very important point to make. Does anyone else have oh, go ahead.
May I just add one thing in terms of, if you're interested in endpoint development for rare diseases, we do have a program, our DEAR program, that you could work with us and to try to, develop the endpoint earlier, and for, hopefully for more than one rare diseases in that case.
Any other questions?
I have a quick question. Todd, you were kind of referring to HIV and to oncology, and that they're fast progressing, and at that time too, there was a lot of regulatory flexibility that was afforded those diseases. Dr. Pazner's done a lot to accelerate that. I'd like to hear your thoughts, and Gumei as well, like what kind of, in the Parkinson's or Alzheimer's space, what do you see in terms of, accelerating these kinds of trials and these long progressing trials, and what kind of regulatory flexibility might we be able to kind of look at.
I'll just- 1 second. So, I mean, the hope is, I think, at least today, is obviously in the space of protein biomarkers. So we now know of protein biomarkers in the blood. We do work in the skin, also in the spinal fluid. And they seem to be fairly sensitive and specific. We don't yet have great examples where we can say, in the work that I do with Parkinson's disease, we can see that the amount of synuclein is elevated in a person with Parkinson's disease. We haven't yet had that therapeutic to say, okay, now the amount of protein goes down and the patient gets better, right? Those are the two things you need to actually have. So we haven't had that therapeutic yet to say that's really going to be the winner. And there certainly is a school of thought out there that says these protein biomarkers are interesting, but are not really the pathological problem. They're not the pathophysiology. They're just an indication of some type of disruption. But I think, I mean, that is where all of the emphasis is for the most part. So most of the therapeutic trials in Alzheimer's are directed at lowering amyloid and tau. In Parkinson's, lowering synuclein. Even rare diseases like, multiple system atrophy and pure autonomic failure, we're trying to lower the protein because we see the protein build up, and we've known that for 100 years, right, in these diseases. So, will that be the winner? I don't know. And then I think the question becomes obviously, and then Alzheimer's is the example, right? So using amyloid PET scans, the anti-amyloid therapies are amazing at taking amyloid out of the brain. But the clinical benefit over at least a relatively short period is minimal. And your point is a really good one, which is, on the one hand, if you have a disease that evolves over decades, then early intervention really allows you to start to think about disease prevention at some point when we're really smart. The downside to a disease that develops over decades is it takes a very long time to show a change unless you really have the biomarker to say, yep, you nailed it, right? And that's your point is a good one, which is, HIV is the great example, but as is diabetes, right? I'm a diabetic. I wear an insulin pump and I have my monitor. I know what my blood sugar is doing every second of every day. I monitor that biomarker every second of every day. That allows us to treat that disease. If we could do that in the brain, we'd be in a very different place, but we're not.
Yeah, I think regulatory flexibility comes in many forms, and obviously you probably all know that we have many programs, expedited programs to help sponsors to develop any treatment either for Alzheimer's, Parkinson's, or for rare disease or pediatric diseases. I mean, their fast track designation, their breakthrough designation, and specifically for gene therapy, there's a regenerative medicine therapeutic designation, RMAT designation. So those programs will, facilitate the development and help you really enhance communication with our sponsors and patient advocacy groups as well to really push the product development to the finish line. And going back to the biomarkers, I think for Parkinson's, for Alzheimer's, for all those chronic diseases that develop over a long period of time, Huntington's, SCAs, biomarker is very important, and we are open to talk about it. We're open up to conversations, and again, show us data, come to us, and we'll, that's a good start point. We'll go from there.
Thank you. And that's actually a really great place. I think, we're gonna have to wrap up for the next session, but that gives me something to just really carry, kind of add a punctuation mark to what you just said, Gumei, because we've, connecting the dots to some of the previous sessions, data sharing is crucial. Understanding what you're measuring it carefully, making sure it's meaningful. All of these are, pieces of the puzzle. And then the last thought I just want to leave with all of you is that clearly it takes a village, it takes an army. I'm so pleased that the panel here represents different segments of the drug development community, and I hope that we gave you some food for thought in mechanism-based drug development. Thank you for your questions and attention.